All right, so anyway, we were in the part of chapter 5 which talks about speeding up and slowing down chemical reactions by affecting enzymes. And we said that there was direct ways of doing that and indirect ways. The direct ways are physical things in the universe that affect proteins and speed up or slow down chemical reactions. When I really said be quiet, I really meant it. Okay, so these are the physical means, and remember the organization of this is from the least efficient to the most efficient of this category. So the least efficient, meaning uses the most ATP for the cell, and this would use the least amount. And we talked about these, and the only one that's really kind of shocking is the one about temperature, and the fact that bacteria are unique in the fact that if you raise their temperature by 3 degrees centigrade or 5 degrees Fahrenheit, it denatures their enzymes and they immediately stop. So if you want to affect a chemical reaction, the quickest way to stop a chemical reaction is to raise the temperature 3 degrees centigrade or 5 degrees Fahrenheit. Okay, another unusual thing that we talked about pH is one of the unusual direct ways of affecting an enzyme. And why is it? Because enzymes work by shape, which is called their nature. And when you put an enzyme into an acid or extreme acid or extreme base, they are denatured. They lose their shape. But pH is the only one where if you quickly move it back to neutral, it will refold into the correct shape and still function without any damage. And that's called renaturation. Now, bacteria actually will renature when they're frozen. And I forgot to mention that. That is one of the ways that we store bacteria is we freeze dry them or we quick freeze them. And if you do this, because they're so small, crystallization of ice particles doesn't cause as much damage to bacteria as it does to eukaryotes. So I can't freeze you, but I could freeze E. coli and bring it back to life. And it might kill still 50% of them, but I would still be able to get the bacteria back alive. Some people ask me, how does radiation affect an enzyme if radiation affects DNA? And remember, the inducible enzymes are coded for by DNA, so if you damage the DNA, then you get the wrong code, you get the wrong enzyme. So that's how radiation works. And remember, there are two kinds of radiation. There's ionizing and non-ionizing. And non-ionizing radiation is UV light. And UV light has some particular characteristics. Number one, it doesn't penetrate glass very well, and it only penetrates cells 1 cell deep. So non-ionizing radiation, UV light, is only effective in killing microbes that cause disease for bacteria and viruses. So UV light is used to sterilize air and surfaces. How does UV light work to destroy DNA and thus the enzyme that it codes for? And the answer is it causes thymine dimers. Thymines that happen to be next to each other on a DNA strand will bind to each other instead across, making messenger RNA. These are called thymine dimers, and since thymine is a pyrimidine base, they're sometimes referred to as pyrimidine dimers. Don't let me trick you by calling it purine dimers. That's the other base. All right. How is this fixed? Because a lot of people want to know, if UV light does this on DNA, then how come we don't get cancer every day when going out in UV light? And remember, there are 3 repair enzymes that we'll cover in chapter 7. And the 3 repair enzymes, the first one and the most effective one is called light repair. And it involves one enzyme called photolase. And photolase is kind of unique in the fact that it repairs UV light, but it requires UV light to be activated. So this enzyme, what it does is it runs across every DNA strand in the cell, and when it comes across a thymine dimer, it pops the double bond there between them, just like popping a zip, and it flips back to normal. So that's the first mechanism of repair, and the best one is called light repair, or photolase repair. The next one involves several enzymes, and it has an awful name called dark repair. And remember, I hate those kinds of naming because that's like saying you're a girl because you're not a boy. You'd have to know what a boy was to understand what a girl is. So it's unfortunate that most people call it dark repair. I call it excision repair because that's what it does. It's like operating. So it's more than one enzyme. They come in toward the damaged area. They see the damaged area. They back up 2 bases and excise the damaged area, cut it, and then resynthesize it using the other strand. And that's called dark repair or excision repair, and it requires several enzymes, not just one. Finally, the third repair mechanism is not very good, and that one is called SOS repair. And it's when a bacteria has been exposed or DNA has been exposed to UV light for a long time, and one area of the DNA strand has thousands of thymine dimers. And so what happens in SOS repair is the cell repair enzymes just cut out the double strand of this whole region and glue the 2 ends together, hoping there's enough DNA for the organism to survive. Usually not. So SOS repair is usually useless. Okay, the other kind of radiation is called ionizing radiation. It's X-rays, solar rays, nuclear rays. And what it does to affect the translation of proteins is that the DNA it either inserts or deletes bases. And that's called frame shift because every 3 letters means something. So if these 3 letters, AAG, means something and stands for an amino acid or something like that, and you can take out this A, then the 3 move down, and now you get the wrong amino acid. So everything after the deletion or insertion is wrong. And that's why frame shift is usually fatal. One of the things I forgot to tell you is that anything that changes DNA is a mutagen, and one of the mutagens that is similar to radiation is viruses. Viruses insert. So remember that viruses can also cause mutations by insertion, and as a result, can result in cancer and do result in mutation. Remember, when a virus attacks your cell, it inserts its DNA into your cell, and then its DNA inserts itself often at random places. And particularly if it inserts itself in the middle of an oncogene, it can result in cancer. Anybody remember what oncogene types there are? Oncogene means cancer gene. No, what I'm talking about is what are a lot of oncogenes hypothesized to actually have been in the beginning? Fetal growth genes. They are genes that are active during fetal development that produce growth factors that cause rapid division that is never repeated in the cell again unless you get cancer. And so when you get to be an adult, you are no longer a fetus. They are repressed. They're still there, but they're turned off because you're an adult. If a virus inserts in the middle of that, it can de-repress a fetal gene, make making an oncogene. So that is one of the viral theories that has been proven to be true about de-repressing oncogenes that were once fetal genes that produce growth factors that cause rapid development. Okay, so we're just reviewing a few of the physical or direct means of control. And now let's get to the real nitty-gritty, the ones that actually work really well, that are very efficient, and that is things in nature that look like the substrate, or shaped like the substrate, will fit into the active site as if they are the substrate, but won't function. And those are called competitive inhibitors because they are fake compounds competing with the substrate for the active site. And we have learned to use them a lot. For instance, penicillin. Penicillin is a competitive inhibitor. And it competes for an enzyme that helps make cell wall material. So when you expose a bacteria to penicillin, it doesn't make a complete cell wall, therefore osmotic imbalance causes it to lyse. So that's how penicillin works as a competitive inhibitor. Remember that if the substance we're talking about is a DNA or RNA base a compound that looks like a DNA or an RNA base but isn't, it's called an analog. And most of our early antiviral treatments were actually fake RNA or DNA bases. So, for instance, AZT, the very first drug for HIV infection, was a fake thymine. So when the virus is taking thymines up to reproduce in your cell, it takes up the fake one. When it does, it makes a virus particle that can't reproduce and can't function, and therefore doesn't work, and the infection does not, exaggerate or become greater in more cells. Remember, the purpose of a virus is just reproduction. It takes over a cell, it tells the cell to make more viruses, uses up all the cell's energy, explodes out of the cell, destroying it, which is what causes our lesions and infection and death, and then the new particles infect new cells, which makes us sicker. So remember, AZT is a fake thymine, acyclovir, which we take for fever blisters, herpes simplex 1, is also an analog, a fake base. All right, so the most successful early treatment for bacterial infections was sulfa drugs. And if you recall, all living things need an essential compound called folic acid for life. We need it in large quantities, so as soon as you find out you're pregnant, one of the very first things they make sure you get in your prenatal vitamins is folic acid, because without it, there's a high rate of spina bifida and other birth defects. So folic acid is essential for all living things, including bacteria. And bacteria make folic acid from para-aminobenzoic acid. An enzyme splits it to folic acid. If you apply sulfa drugs to a bacteria, it will competitively bind to the enzyme. Sulfa will. It looks like para-aminobenzoic acid. It's shaped like it. It will occupy the enzyme, but it won't make folic acid, so the bacteria dies. And the reason you don't die when you take sulfa drugs is we don't make it. We eat folic acid. We don't make it from another compound, so we don't have this enzyme to occupy.
Did you say that the bacteria creates folic acid?
They make it from para-aminobenzoic acid. Okay, so that's competitive inhibition. And again, we're going to move to another name that I hate, and that's non-competitive. I would much prefer that non-competitive inhibition would be called allosteric inhibition. So they tell you that instead of competing for the active site, it's a compound that sticks to the allosteric site. Remember, when anything sticks to an allosteric site of an enzyme, it warps the catalytic site, and the substrate won't fit. So things that stick to allosteric sites are called non-competitive inhibitors or allosteric inhibitors. Remember, if they stick to an allosteric site irreversibly, they're poison.
So,
It poisons the enzyme, denaturing it forever.
so you have like a, what is it called? That was labeled when you guys started writing will you pick up these in there?
Actually, what they're doing, they're giving you antivenom.
Yeah.
Antivenom is actually a stolen antibody from another animal. It's passive immunotherapy. What they do is they go out and get snake venom. They go and milk a snake. They take the venom and they inactivate it a little bit and they inject it into a horse. And the horse makes antibodies, which are called antivenom. Then they treat the blood, they drain the blood, some of the blood from the horse, and they purify that antivenom and stick it in a refrigerator so that when you get bitten by a snake, you can use the horse's antibodies. It's called passive immunotherapy, and it's stolen antibodies because there's no time after a snake bite for your body to make its own antibodies in time to save. Sorry?
What happens to the active enzyme that these allosteric sites reversible?
Oh, you're talking about poison. I mean,
That's not the same thing.
yeah, not the same thing.
That's not the same.
No, it is poisonous, but it's not a poison. A poison is something that sticks irreversibly to an enzyme, a necessary enzyme. And remember, when it sticks there, it denatures the enzyme
Right.
permanently. We talked about carbon monoxide.
Yeah.
Does it warp the enzyme substrate cofactor?
Nope, it warps the catalytic site. It changes the shape so it won't fit. Now, the cell uses allosteric inhibitors that are reversible to control enzymatic reactions. And the most famous one being feedback. Feedback is the best of all our direct enzymatic control mechanisms, but all of the indirect are better than any feedback. And feedback is when you have a series of enzymes working together to get an end product. And what the cell does is it uses its energy to make this end product, but it doesn't need all of it all the time. It just needs a constant supply. But what it does is it fills the entire cytoplasm with the end product until by chance the end product sticks to the allosteric site of the first enzyme. Once it sticks here, there is still more end product in the pipeline, so the cells make a gluttony or a huge excess amount of this end product. Waste ATP, making lots of end product when it only needs a constant supply, not an excess. So what happens? The pathway is blocked, and then each time the cell will use some of the end product that's in the cytoplasm until all of it's gone, and then it pulls the last one off and uses it, but it's got to wait until more is made. So you have, stuffing yourself and starving, stuffing yourself and starving, which is not an efficient way of controlling reactions, but it's better than nothing. Okay, so that takes us to the hard stuff. And we've got to do a lot of preparation before we actually explain it. First of all, again, indirect or genetic control of enzymes is organized from the least efficient to the most. So which one it uses the most ATP of these methods? Enzyme induction. And which one uses the least? Catabolic repression. All right. So then we look at the words. And we want to know, what is the cell's natural condition before this control mechanism kicks in? So, on enzyme induction, what is the cell doing before this kicks on? Is it on or off? Induction.
Oh.
It's off. Its normal condition is off, and this mechanism induces it, turns it on. Okay, let's look at the second one. What is this cell normally? on or off. It's called enzyme repression. To save ATP, it turns it off. It's normally on. So that you need to know. Next, you need some vocabulary. So we're going to write down some vocabulary terms, and the first one we're going to write down is an operon. And these are my favorite definitions, so, you know, it's good for understanding, and so they are overly simplified, I understand. What is an operon? A series of control genes working together to make a particular protein. A series of 4 control genes working together to turn on and off the synthesis of proteins. A series of genes working together to turn on and off the synthesis of proteins. That's an operon. Anyone need it again? Okay, next. An operon has 4 parts. And remember, in genetics, a strand of genetic material, anything that goes to the left is said to be upstream, and to the right is downstream. So it's just vernacular, okay? So, of the operon, the farthest upstream is the regulator. And it's abbreviated little I. The next part of the operon downstream is the promoter. Following the promoter is the operator. And finally, the important part of the gene, the one that actually does the work, are the structural genes. So, the operon's genes are regulator gene, promoter gene, and operator gene, followed by the ones that actually make the enzyme, the structural genes. So, this is how we're going to start. So let's look at this one and remember what we're talking about. This was discovered in E. coli. E. coli doesn't digest lactose very often because it never comes across it. So the genes to turn on lactose, to make 3 enzymes to digest lactose, are normally turned off because E. coli doesn't see lactose very often. But it has basically an emergency channel. If it runs out of food and lactose is available, it can induce the production of the 3 genes that digest lactose through this inducible operon. It's on their DNA, but it's not normally read, because every day, remember, this is turned off. Alright, so this is the enzyme induction, and remember, it's normally off. So every day, here's what happens. Up here at the regulator, every day, 365 days a year, 24 hours a day, and 365 on a leap year, the regulator is making repressor protein. So what's it going to be doing at 10 p.m. tonight? Making repressor protein. What was it doing at 8 a.m. this morning? Making repressor protein. All right, so when it makes that repressor protein, notice the shape of it. It fits right into the operator. So here's what happens. Here we have a strand of messenger RNA. This is what this is, messenger RNA. And what happens when there's a strand of messenger RNA? A ribosome translates it, right? So here's what happened. A ribosome comes along here, and when it sees a promoter, that promoter basically is a signal that says attached to the messenger RNA here. It moves forward, and when it gets to the operator says to the ribosome, everything after this should be translated. But the repressor sticks there, just like driving a car across the tracks of the blue line. What happens to the train? It stops. If you have a tree across the track, what's the train going to do? Stop. So here's what happens every day. Repressor is made, repressor sticks to the operator. Ribosome comes along the messenger RNA, hooks in, gets here, finds it blocked, unhooks, and goes to the next promoter, skipping everything there. That's what happens every day. It's off. All right, so what happens if lactose gets in the cell? If lactose enters the cytoplasm, then lactose preferentially will bind to the repressor. So as lactose floods into the cell, all the existing repressor, and the repressor is very fragile, so it's made and it falls off, and it's made and it falls off. All of them are occupied by lactose. As they're made, they're occupied by more lactose. When the repressor is not sticking here, ribosome comes, hooks, goes on, and it translates the Z structural gene and makes beta-galactosidase. It translates the Y gene and makes beta-galactosidase permease. And then it translates GA and makes transacetylase. Now, what is lactose? It's a disaccharide. So what do these enzymes do? Beta-galactosidase breaks lactose into galactose and glucose. Beta-galactosidase permease assists lactose through the plasma membrane. So more lactose is flooding in. And transacetylase, we think, no one's absolutely certain yet, we think it has something to do with the processing of galactose, but nobody's sure. All three of these are needed for lactose to be digested. There is no lactase. People have memorized for years that lactase is the enzyme that breaks down lactose. It is not. It takes three.
All three are needed for what?
All three are needed for lactose to be utilized as a substrate. And it's an inducible substrate because the enzymes are not a part of the plasma membrane. They're only made when they're needed. And when are they needed? When lactose floods into the cell. Now here comes a question that requires thinking. All right. What de-represses the lac operon? Now remember represses means it's off. De-repressing means it's on. Remember in English, off is on. No is yes. If I tell her, get over there and off the off light, what does that mean? Off the off.
Turn it on.
Turn it on. So remember, if you're being raped, do not scream, no, no, because no means yes in English. In Spanish and Latin language, no damn well means no, damn it. But not in English, no means yes. So be very careful what you're screaming. All right, so what offs the off operon? Lactose offs the off. What turns it off? The repressor. What offs the off? Lactose. So what de-represses its own operon? The substrate, lactose. Everybody got it? Not bad, is it? Not bad at all. All right, so if you got that one, the next one's not going to be bad. All right, the next one is repression. And this one is the trip operon. And notice how much ATP. Does it take more ATP to run the trip operon or less? Look at how many structural genes we got. We got 5. That's a lot of ATP. And remember, this is running all the time because its normal condition is on. Maybe I better add that there so everybody remembers. Oh, yes, it says it's normally on. Why? The cell needs tryptophan every day, all day long, constant. So it makes it. And it takes a lot of ATP to make it. So do you want to make extra? No, you don't want to make extra. So every time one is made, it's used. So here's the story of how this works. Notice, what's the regulator doing 24/7? Making repressor. But will the repressor in this shape fit to the operator? No, it won't. So normally, it won't fit here, so this is running all the time, even though repressor is made. All right, so anyway, when I hope my mother never sees this. All right, when I was a child, I had polio at age 2, and then I was in the hospital for like 2 years, and then like 15 operations later, and moving muscles so I could walk. My father, I didn't find this out till I was 16-year-old. When you're 16, what do? You go through all your parents' things. When they're not there, you go through them. You find the things hidden in their bedroom. You find the things in the sock drawer, and you wonder what mom and dad are doing with those. And then you go through all their paperwork. Y'all didn't do that? Oh God, I did. I went through all the files, read everything, make sure. Yeah, my dad's a scientist. He has all these, filing cabinets, and I went through everything. I hid it. I put everything back so they would never know. But one of the things I found was my father was still paying at 16 for my operations. And I didn't know. I went and asked my mom, and she said, well, your granddaddy was rich, but when my dad went to ask granddaddy if he would pay for this unique surgery you needed to move muscles so that you could walk, because remember, polio kills the nerves that keep your muscles working, and if they're not working, they fade away, they atrophy. So polio kills those nerves that activate the muscles, so the muscles atrophy. So after you get over polio, you're missing some muscles that would cause me to be able to walk. And so this famous Jewish doctor in Atlanta, Children's Hospital, Crippled Children's Hospital, had a new technique where he would move muscles from one area of the leg to others so that you could walk, to give you minimum. So they moved these muscles, and these operations were horribly expensive, hundreds of thousands of dollars. And my father went to my grandfather and said, you're rich, would you help me pay for this? Maybe advance me the money and I'll pay you back with interest. And my grandfather thought about it for a while, and he was a big businessman, and he said, it's not a good investment.
Oh my gosh.
I know, my father never quite got over it. He tried to forgive, but I could always tell there was that little look, kind of thing. And of course, my mother was devastated. And so my father actually went and borrowed money for the surgeries, and he didn't pay it off until I was 18 years old. He paid all those years. And I remember looking at his check, and it was $600 a month. I wonder how he raised a family of 4 on $600 a month, built a house and had a car, and paid off this. He must have been paying like $15 a month or something. But anyway, after the surgery, he was desperately afraid I would be gay. Little did he know it wasn't much environment, it was mostly genetics. But my mother's side, it's on the female side. But anyway, he decided that at age 9, he was going to make a man out of me. And the only way to do that is baseball and softball, the American sport. So he said, we are going to learn to play softball. I'm going to go buy you a catcher's mitt, and me one, and I'm going to buy us some balls and a bat, and we're going to practice, and you're going to love it. But never mind. So we played catch, and I got better and better at it, and I was hitting, and okay, and all that. And he said, now we're even going to have more fun. We're going to go to a batting cage. And they have a machine there that will pitch. So first you'll learn to catch, and then you'll learn to bat. And we can set the machine on slow, so it won't be a problem. Then we can set it on faster and faster, so you get used to it. And then we'll set it on intermittent, so you won't know what's coming. And so we did it, and the machine's pitching it, I'm catching it, laying the ball down, catching it, laying the the ball there. That's what the cell is doing every day. It's making tryptophan, synthesizing. The moment it's made, it's used. The moment it's made, it's used. But like me, my father put it on high-speed intermittent. And that meant, boom, one goes over my head. That's what happens to the cell. It's regularly using it, but it gets out of sync and it makes more tryptophan than the cell can actually use at that moment. It makes one extra. And when it makes one extra, tryptophan is a co-repressor. It will stick to the allosteric site of the repressor, altering the shape so it now fits at the operator, blocking excess synthesis. So when it gets one extra, it will stick to the repressor, and the repressor will stick to the operator, and then, but remember, there's still one in here. So actually, how many extra are there? 2, 3. But the system is stopped, so it uses that one that's in the process of being made, and then it takes this one off. This flips back to that bad shape, and the system goes on. So it's boom, 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 boom. Stop. That one's used up. This one's pulled off. Boom, boom. It's a way of regulation to keep a nice, consistent, and nothing extra. But this one, how many extra do you get when you get extra? You get 2. One goes over the head, and it stops it, and then there's one in the passageway by the time it stops. Okay, so anyone not understand enzyme repression? Tryptophan is a co-repressor. So when there's an extra one, it sticks to the repressor, altering the shape, which sticks to the operator just like the lac, and blocks further synthesis. No waste, well, very little waste of ATP. You're not like feedback, filling the whole thing up before you turn it off. Okay, no one has any problem with that one? Let's move on then to attenuation. Anybody know what the word attenuation means? Fine-tuning. In the old days when you had those old FM radios where you didn't have push buttons, you turned the dial. And when you got to, 98.7, you would have to turn it a little bit forward, see if it went out, and a little bit back to see if it went out, and then you get it right in the middle and you get the strongest sound, right? That's what this is. It's fine-tuning. Remember that making an excess? Guess what? The second enzyme in the pathway, 2 places on the third and 1 place on the fourth, has allosteric sites that tryptophan can stick to. So instead of stopping it here, you can stop it where 4/5 of one. So 1 extra is made, it sticks here, and you get, instead of getting 2, you get 1 plus 4/5 of 1 made. So the 1 is then used, and then this falls off, and now you immediately have 1. The cell doesn't have to wait as it does if it's back here. So this is fine-tuning.
It does both?
Yeah, but this one's a different, it's the same operod, 2 different ways it's controlled. One by repression and one by attenuation. Attenuation is where the product will fit to the allosteric side of enzymes 2, 3, or 4.
Right.
And if it sticks to 4, then you only have 1 and 4/5 of 1 extra. The moment you use the 1 up, immediately you have the next one. So there's no lag time or wait for the cell to get the tryptophan it needs constantly. So this is a better method than the last one. Anyone have trouble with it? Told you. If you understood the lac one, you've got the rest of them there easy. All right, the last one, and explaining it is going to be incredibly easy because nobody knows. And this is the one that says that somehow, these are called fantasies. Okay, or as my psychologist once said, magical thinking. You guys know what that is, right? Where magical thinking is where you think, well, I don't need to fix my own psychological problems because there's someone out there that can fix me for me. So I don't have to work on it. there's always somebody out there. It's called magical thinking. Trust me, you have to fix you. There isn't anybody out there that can fix you. That's too passive aggressive. All right, so anyway, I told you I was a psychology major. Here's what happens. When a cell, like E. coli, is put into the substrate or a dish where both a constitutive substrate and an inducible substrate exist at the same time, the plasma membrane forbids the inducible substrate from passing through. We have no idea how the plasma membrane controls lactose passing through it. All we know is, if you put E. coli in a plate with both glucose and lactose, it will eat all the glucose, and the plasma membrane is impermeable to lactose until the glucose is gone. And suddenly, it's sort of like red light, green light. And then, remember, when you get the enzyme permease, it helps even more through. So, catabolic repression is the control of the permeability of the plasma membrane. And the plasma membrane is not permeable to a inducible substrate until the constitutive substrate is gone. It, just like a door shut, says no lactose in, there's plenty of glucose. Remember the old analogy I gave? When you enter the house and you have your favorite pizza and fried liver, which one are you going to eat? The pizza. Trust me, all the pizza, and then I'm going to starve before I have to eat that other crap. Don't say anything, Mom. No, she loves it. And of course, the dioxin growth curve that we talked about before.
So we don't know how it happens, just know that it happens.
We know it happens. We don't know how the cell membrane does it or what signals it or anything about it other than we observed it happening. Okay, questions? That's chapter 5. The next one is a fun chapter, not too hard. And it's chapter 6, growth. I don't like this one. See if there's another. Hold on. You're so far ahead of the other class, haven't done it yet. All right, so the other day when you guys first began reading your tubes, I had a whole bunch of people, I wish we had the lecture then, because I wouldn't have got the question, but I had a whole bunch of people come up and say, look, at the bottom of my liquid, there's some stuff there, what's that? And I had people, oh, look at the top, it's all fuzzy. Does that mean it's contempt? Growth equal cloudiness. Okay? In bacteria, the more it grows, the cloudier the liquid gets. And so when you see cloudiness, you know your bacteria are reproducing. I had, sadly, I used to tell this story, she was still alive, last year she died. I had this 99-year-old aunt. Incredible person. You know, some people in your family, so listen to this moment. Some people in your family, you're stuck with. You know, they're related to you, there's nothing you can do about it. And you wish you weren't, but that's the way it goes. And then there are others you go like, how did that person get in my family? Incredible. They're just unbelievable. And you say, nobody else is like them, why can't we get more of those? So my Aunt Ruby was an incredible person. First of all, at 15, she met her boyfriend and he took her out of Alabama. So she left Alabama at 15 and went to Chicago. And she always had a lot of drive. She didn't want to be a burden on any man. And so she went. There are only two options for women in those days before, World War I. And her option was to be a nurse or be a secretary. So she went to secretarial school. That was it for women, just about. And so in Chicago, she went to secretarial school and she got a job with Sears and Roebuck. And by the time she was in her early 60s, she was executive assistant to the president of Sears and Roebuck Corporation, and she worked in the Sears Tower at the time, the tallest building in the world. And she was extremely successful, and then her husband died. And she continued to work till she was 63 when she could retire. And weirdly to me, she returned to Alabama. We never saw her before that. She just came back after she retired. I asked her one time, Miss Ruth, why did you come back to Alabama? After all, you're a Notre Dame fan, you have a Yankee accent. What are you doing here? And she said, $350 a month. I said, what? She said, well, an apartment in Chicago is $800 to $1,100 a month. Even a studio is $350 a month in Andalusia for a 2-bedroom. It's so much cheaper to live down here. And I said, and then she said, and have you, they don't call Chicago the Windy City for nothing. It's cold and it's windy. And it's just not good for old people. So I moved back to Alabama. Well, what's so unusual about Miss Ruth was that I never saw her when she wasn't perfect. You didn't drop by her house. She was a Yankee. In the South, anybody will drop by. They don't call, they don't text, nothing. They just drop by. And they walk in the back door and they sit down and they pull up. My mom will hand a plate and suddenly they're having dinner with you. Not Miss Ruth. You don't go to her house like that. You call a day in advance. And nobody ever saw her in her PJs or anything like that. If you saw her, she was perfect. Her makeup was perfect, and she never wore a dress. I never even saw a dress. I said to her once, Miss Ruth, why don't you wear dresses? And she said, men don't take women seriously that wear dresses. She always had a suit and a different brooch every day. And she was 5'1", so she wore stiletto heels until she was 95. And I said, why do you wear stiletto heels? It must kill your feet. And she said, the hell with my feet. No man's gonna look down on me. I'm looking him straight in the eye. She's a tough old broad. Amazing person. I never once, ever heard her say anything bad about anybody. Ever. And in Alabama, with all the gossip that goes on and all those religious people that are, she never, ever. She was the one that brought that old saying in the family, if you can't say something good, don't say anything. So she was an incredible person. And every time she saw me, remember I was 240 going to 270 pounds. So every time she saw me, she'd grab a huge chunk and shake my head around like this and go, my God, you're getting to be a big old thing. Just more of you to love every day. Now, you can't say to your 99-year-old, that hurts my feelings. You just say, oh, Miss Ruby. She knew that there were more cells of me. Love, that's growth. Cells don't get bigger, particularly bacterial cells. They have a cell wall. They don't get bigger. So if you feed them, there's more of them. So remember, growth equal an increase in number, not an increase in size of cells. So you have growth equal cloudiness equals an increase in number of cells. It is not an increase in size. And cells in bacteria divide by binary fission, which is exact copy division, mitosis. And remember, there's only basically 2 stages. We don't even say there are 2 phases. We say, you know, it's just called binary fission. But essentially, it's telophase and interphase. And in interphase, nothing appears to be happening, but DNA and cellular parts are duplicated. Now, the time it takes for one cell to become 2 is called generation time. And in bacteria, it's extremely short. It can be as short as 20 minutes. And they actually found one that's a little over 60 seconds. So generation time in bacteria is extremely quick. What is generation time? The amount of time that it takes for one to become 2. Generation time is distinct among all species. There is a book you can get that tells you the generation time of every species, and every species is different. So you can look up that E. coli is, I think, 61.25 minutes, something like that. If it was easy to measure, you could use that to identify your bacteria, but it's not easy to measure where one generation ends and the next one begins. So generation time, even though it's unique to each species, is not easy to measure. All right, so now we get to the growth curve. Remember our dirty little secret. Close your eyes. Imagine everything you ever wanted in one little spot, in one little room with a plexiglass wall around it, this size. And if you did, imagine everything you ever wanted. What would be the absolute perfect thing? Remember, I told you mine. It would involve sex, beach, and chocolate. Maybe a mixture of all of them. All right? So, when you open your eyes and you're there in heaven, or the optimum of everything, what's your first reaction? Remember? Shock. All living things go through the growth curve. And when you're dropped into a limited environment under optimum conditions, that period of adjustment is called lag. Lag phase of the growth curve. And in bacteria, it's usually about 2 hours. So remember, lag phase is a lot like prophase. Nothing appears to be happening, but internal parts are duplicating, and the cell is becoming adjusted to these optimum conditions. And then the cell's metabolic rate moves to its maximum efficiency, and the generation time becomes the shortest. And this is called log or exponential growth. Phase of the growth curve. Okay, and remember that in log growth, all enzymatic systems are at maximum efficiency. The shortest time between any 2 replications is defined. So generation time is defined in log growth. And just like if a person is put into optimum conditions, it is where they are gobbling up everything and reproducing as fast as possible, as fast as that organism is capable of. Its maximum capacity of everything. And that's log growth. And what's important about log growth? If I ask you which of the phases of the growth curve is the most important, the answer is log exponential growth or log growth. Why? 1, generation time is defined there. 2, everything is at optimum. Maximum efficiency and optimum. And finally, all references report results, assuming that the inoculum was taken from a bacteria in log growth. understand why in a moment. So this is what makes log growth the best. And remember, the most important, it's exponential growth. 1 becomes 2, 2 becomes 4, 4 becomes 16. Each time it squares. Now what's happening in the limited environmental system that we have? Remember, this is limited. You can't get out of these conditions, like a plexiglass dome or whatever. Nutrients are being utilized at an excessive rate. If it's aerobic, oxygen is being used up at a tremendous rate. And remember, everything that is metabolizing is also pooping. So waste are building up at an amazing rate. And remember, if it's reproducing, overcrowding. You know, when people become overcrowded, what do? They get psycho, they stop reproducing. When you get a heavily populated area and you can barely move, people get very annoyed easily, they get very short-tempered, they do violent things, and they certainly don't feel like reproducing. So that's what happens in log growth. You're getting huge amounts of population increase, huge decrease in nutrients and oxygen, and huge amounts of waste. And as the things build up to about here, then the number being reproduced equal the number dying. And that's called stationary. And the reasons for stationary, who can tell me? Why does the population level off? Too many crowd, too many overcrowding. I mean, you know, how do you have sex when you can't move?
They're dying.
But there's no more food. You know, there's lots of dead bodies around. You know, you're walking through 6 feet of dead bodies. What else? No more nutrients. Nothing's coming in, so they're getting all used up. So the population, the number being reproduced, will end up equaling the number dying. That's stationary. And the most important, interesting thing, besides the population being equal, being reproduced equal to number dying, call it ZPG, zero population growth, is that somewhere during stationary, and we don't know where, somewhere during stationary, and always during death, a very unusual event takes place called involution. Involution is a word that means upside down, and it refers to the results like gram stain, acid-fast, endospore, and your biochemical reactions like phenyl red glucose and phenyl red lactose and all those. All the results that if you take an inoculum from stationary, remember the waste have eaten a hole in the cell wall and damaged the plasma membrane by then. The acids from the waste. So if you take that bacteria and inoculate it into one of your test reagents, or you do a gram stain on it, it's not going to come out right. That's what involution means. The results are upside down. Gram negative stain gram positive stain gram negative. Phenyl red glucoses are supposed to be positive, but they're negative. There are all kinds of weird results. And that is why all experiments are assumed your inoculum came from here. Log growth, because of involution. That's why when we first did our isolation and you took your little plate and you streaked it and we put it in the incubator overnight, and then a couple of days later we came back and you did the gram stain on the dots, and everybody I told people, we're going to do a gram stain, but we're most interested in size and shape. We're not so interested in gram stain because the gram stains will often not be right. Why? Because if this is 2 hours lag, then thousands of thymine dimers. And so what happens in SOS repair is the cell repair enzymes just cut out the double strand of this whole region and glue the two ends together, hoping there's enough DNA for the organism to survive, usually not. So SOS repair is usually useless. Okay, the other kind of radiation is called ionizing radiation. It's X-rays, solar rays, nuclear rays. And what it does to affect the translation of proteins is that the DNA it either inserts or deletes bases. And that's called frame shift because every 3 letters means something. So if these 3 letters, AAG, means something and stands for an amino acid or something like that, and you take out this A, then the 3 move down, and now you get the wrong amino acid. So everything after the deletion or insertion is wrong. And that's why frame shift is usually fatal. One of the things I forgot to tell you is that anything that changes DNA is a mutagen, and one of the mutagens that is similar to radiation is viruses. Viruses, and when you put an enzyme into an acid or extreme acid or extreme base, they are denatured, they lose their shape. But pH is the only one where if you quickly move it back to neutral, it will refold into the correct shape and still function without any damage. And that's called renaturation. Now, bacteria actually will renature when they're frozen. And I forgot to mention that. That is one of the ways that we store bacteria is we freeze dry them or we quick freeze them. And if you do this, because they're so small, crystallization of ice particles doesn't cause as much damage to bacteria as it does to eukaryotes. So I can't freeze you, but I could freeze E. coli and bring it back to life. And it might kill still 50% of them, but I would still be able to get the bacteria back alive. Some people ask me, how does radiation affect an enzyme if radiation affects DNA? And remember, the inducible enzymes are coded for by DNA, so if you damage the DNA, then you get the wrong code, you get the wrong enzyme. So that's how radiation works. And remember, there are 2 kinds of radiation. There's ionizing and non-ionizing. And non-ionizing radiation is UV light. And UV light has some particular characteristics. Number 1, it doesn't penetrate glass very well, and it only penetrates cells 1 cell deep. So non-ionizing radiation, UV light, is only effective in killing microbes that cause disease for bacteria and viruses. So UV light is used to sterilize air and surfaces. How does UV light work to destroy DNA and thus the enzyme that it codes for? And the answer is it causes thymine dimers. Thymines that happen to be next to each other on a DNA strand will bind to each other instead across, making messenger RNA. These are called thymine dimers, and since thymine's a pyrimidine base, they're sometimes referred to as pyrimidine dimers. Don't let me trick you by calling it purine dimers. That's the other base. All right. How is this fixed? Because a lot of people want to know, if UV light does this on DNA, then how come we don't get cancer every day from going out in UV light? And remember, there are 3 repair enzymes that we'll cover in Chapter 7. And the 3 repair enzymes, the first one, and the most effective one, is called light repair. And it involves one enzyme called photolase. And photolase is kind of unique in the fact that it repairs. All right, so anyway, we were in the part of chapter 5 which talks about speeding up and slowing down chemical reactions by affecting enzymes. And we said that there was direct ways of doing that and indirect ways. The direct ways are physical things in the universe that affect proteins and speed up or slow down chemical reactions. When I really said be quiet, I really meant it. Okay, so these are the physical means, and remember the organization of this is from the least efficient to the most efficient of this category. So the least efficient, meaning uses the most ATP for the cell, and this would use the least amount. And we talked about these, and the only one that's really kind of shocking is the one about temperature, and the fact that bacteria are unique in the fact that if you raise their temperature by 3 degrees centigrade or 5 degrees Fahrenheit, it denatures their enzymes and they immediately stop. So if you want to affect a chemical reaction, the quickest way to stop a chemical reaction is to raise the temperature 3 degrees centigrade or 5 degrees Fahrenheit. Okay, another unusual thing that we talked about pH is one of the unusual direct ways of affecting an enzyme. And why is it? Because enzymes work by shape, which is called their nature. There's UV light, but it requires UV light to be activated. So this enzyme, what it does is it runs across every DNA strand in the cell, and when it comes across a thymine dimer, it pops the double bond there between them, just like popping a zip, and it flips back to normal. So that's the first mechanism of repair, and the best one is called light repair, or photolase repair. The next one involves several enzymes, and it has an awful name called dark repair. And remember, I hate those kinds of naming because that's like saying you're a girl because you're not a boy. You'd have to know what a boy was to understand what a girl is. So it's unfortunate that most people call it dark repair. I call it excision repair because that's what it does. It's like operating. So it's more than one enzyme. They come in toward the damaged area. They see the damaged area. They back up 2 bases and excise the damaged area, cut it, and then resynthesize it using the other strand. And that's called dark repair or excision repair, and it requires several enzymes, not just one. Finally, the third repair mechanism is not very good, and that one is called SOS repair. And it's when a bacteria has been exposed or DNA has been exposed to UV light for a long time, and one area of the DNA strand has thousands